to chair this session. I'll kick off with some introductions and some housekeeping for the next hour. Just to say that this webinar is recorded, so as well as the people watching it live tonight, there'll be many people who are watching it later on as well. So, I'm Azza Faruqi. I'm the Clinical Director of the National Clinical Research Network in the East Midlands for the NIHR. The NIHR is the National Institute for Health and Care Research, and it's a body that organises research within the NHS. I personally have been part of the NIHR for more than 10 years, and I've been the clinical director for the last year and a half. My own clinical interests, I'm a GP. I also have a clinical interest in diabetes and research in diabetes. As well as that, I have interest in research in ethnic minority health. But tonight's about genetics. So I'm going to be putting your questions to Dr. Julian Barwell, who is consultant clinical geneticist at the University Hospitals of Leicester and honorary professor in genomic medicine at the University of Leicester. A big thank you for the excellent questions you've already sent in. If you want to ask a question during the event, then please post it in the Q&A box, and I'll try and pick it up and address it during the next hour or so. The Q&A box is in the speech bubble at the bottom right of your screen. Please ensure your camera and microphone are turned off. That's the buttons at the bottom of your screen. To change how the speakers appear, click the three buttons at the bottom of the screen next to the red phone icon, then change layout, and then select the sidebar or spotlight setting. Unfortunately, we won't be able to answer all your questions about individual cases. However, we will try and answer most of the questions that have been put to us tonight, and some of the ones about individual clinical areas we'll try and answer generically. If you're interested in learning more about research and research for studies you can take part in, then please be part of the research website. Just research, be part of research. Remember, you can take part in as many studies even if you don't have a health condition yourself. So we're now going to go to the first question over to Julian. And the first question is, Julian, where is the best research into the genetics of schizophrenia going on?
Thank you so much, Azar, and for the opportunity to speak tonight at this event. It's a huge honour to be asked. So, obviously, something like schizophrenia is incredibly complicated to unpick. So we've got 20,000 genes, and our oldest gene is actually 2 billion years old. But we have so many environmental and psychological factors to take into consideration when thinking about something like schizophrenia. It can be very difficult to unpick the impact of just one gene on all of these other factors. I remember being at medical school back in the 90s, and even then learning a list of 25 different causes of schizophrenia. So the classic way in which we work out the inherited and the heritable impact of a condition is to look at twins. So we look at identical and non-identical twins that are brought up together or apart. The other way of doing it is looking at families where something like schizophrenia is really common, or in a particular syndrome. That's when there's a combination of different types of problems which seem to be occurring together more often than not, and more commonly than you would see in a general population. With regards to the best research which is coming out now, perhaps it's really through the UK Biobank, which looks at several 100,000 patients and looks very broadly at their genome. And what it does is it compares people who have schizophrenia, and perhaps 2 or 3% of the general population have that, and then compares the genomic, the subtle changes throughout our genome, and we all have one of these subtle changes perhaps every 1,000 letters of our genome, and we've got 6 billion overall, and makes comparisons between people with and without schizophrenia. So the best research really is coming out of, I would say, the Maudsley down at King's, linked into King's in South London. And they're doing a lot of very complex mathematics trying to underpin really what's going on. But I think what I learned in the 90s was probably too simplistic, because genetics can impact on behavior in lots of ways. And for instance, one of them is genetics may partly influence where we end up living. We know that migration and moving into city areas seems to be associated with an increased risk of schizophrenia. But it does seem that genetic factors even underpin where we end up living. And some of the decisions that we make in life that may not be directly linked to schizophrenia, but maybe linked indirectly. So I think it's probably too simplistic to say there are environmental factors and genetic factors. They probably interplay in lots of different ways. And so I think there's a lot of interesting and surprising new connections that will be made. But it's classically, schizophrenia is classically not what we call a Mendelian trait, which means it's unlikely that just a single genetic factor imparts a loss of the risk. I would consider schizophrenia similar to how tall you are. It's a combination of some environmental factors, some hormonal factors, and we know sleep, for instance, is impacted by how much you sleep. How tall you are is impacted by how much you sleep at night and how much growth hormone, but also factors, genetic factors that you inherit from your parents as well. So it's certainly a complex trait in the vast majority of cases. And I think genetics will have an impact in lots of different ways that we didn't even think were possible in the past, and didn't even surmise that. So I think the answer is going to be big data sets, and really looking what I would call agnostically at the data. And that means forgetting this idea of having a very clear hypothesis where we say, well, I think this gene is impacting on this risk by this mechanism. I think we need to be looking at bigger data set, bigger numbers of patients, but allowing the computers to do the hard data work and seeing what comes out. And we might find some very interesting new connections that we weren't aware of before, perhaps by machine learning in the future.
Great, thank you, Julian. It's obviously a complex area with the interplay of the genes and your environment. Perhaps I can bring in a question alongside the answer that you just gave around health inequalities. We know that schizophrenia does affect different groups differentially, whether it's your socioeconomic factors or maybe ethnicity. Do you want to say a little bit about how genetic research can help reduce health inequalities or the risk that it may increase them, and what we should be aware of there?
So there's a number of issues there. So the first one is people's awareness and access to genetic testing and genetic research. But then there's another disparity about individuals' ability, not even when they're actually in front of a doctor and undergoing genetic testing, to get a meaningful result. And I think this is partly due to understanding what normal, whatever normal means, DNA in an unaffected population really truly looks like. There's been very little research in all different types of populations to truly understand biology in individuals from different parts of the world. And that means that the ability to make a diagnosis from a genetic diagnosis, already you've got half the power in individuals from Africa to make a molecular diagnosis, even if you've got their blood in the lab, because we just don't understand what genomic variation means in individuals from these different parts of the world. And I think there's a lot of reasons for that. It's partly where the research labs are and ease of access. But I think the critical word here is trust. There's been a lot of mistrust in communities about the misuse of data and the misuse of genetics, which goes back for 100 years and certainly goes back to concerns regarding eugenics and the misuse of data and consent and manipulation of data. So I think we need to build these relationships with communities, and that involves reaching out into communities, so meeting people in their environment, being aware of the cultural and the language issues which are involved in that, and also designing projects with the community itself, design, develop, and have real true patient voice throughout that patient journey, from the conception all the way to the giving the results to studies. Because without that trust and without that relationship, I think people will be very worried about what happens to their data. You see, if somebody steals your bank account details, you could always change your bank account tomorrow. But with your genomic data, because genomics is such a part really nowadays of our identity, it would be very difficult to change that. And as a result, people are naturally very nervous about how their data might be used, both in terms of their identity, but people have concerns about their data being misused, for instance, for things like in insurance moving forward, or if their data is going to be sold. So I do think it all comes down to having that better relationship with communities, having community leaders on board, community champions as well, but also thinking about how we design projects so that they become useful for the entire community that we work with. And I think that's a huge challenge. But without having the people take part, for instance, we do a lot of work in prostate cancer. And we know that people are at much higher risk if they have African descent. But unless people come forward, it will be very difficult to answer those questions, and we'll just continually, unfortunately, lose people from those communities, and as they almost die in silence, really.
Thank you, Julian. Those are really, really important points. And I think we both agree it's really important that we're as inclusive as possible in our research, in all research, but particularly genetics research, precisely for the reasons that you've outlined. Let's move to our next question, which is around infant mortality. Can you tell us about the applications of genetics research in reducing infant mortality, and particularly from congenital abnormalities?
Thank you. So there's a lot of different causes of congenital abnormalities, and it'd be wrong to say all of them are genetic. So we would say perhaps 40% have either a single gene cause. So would be a condition that potentially could run in the family, or be where both parents are carriers of a condition or unaffected. But when they come together, there's a chance that both parents could pass on a gene change causing a problem. So that's called a recessive trait. But it may be a new dominant trait as well. And these sorts of problems, if you add them in with chromosomal issues, probably account for about 40% of congenital abnormalities. So chromosomes contain all of our genes. They're a bit like the old encyclopedia, and we've got a complete set of genes on these chromosomes which are stored, which we get from mum and from dad. So we've got 46, so that's 23 pairs. So chromosomal problems, a lot of people have heard of things like Down syndrome, for instance, where you get a whole extra chromosome 21, and that can cause problems such as congenital heart disease, which you might pick up in pregnancy, for instance, with the scans. And a lot of these problems, like short, long bones, or problems with major organs and limbs, are picked up at those pregnancy scans, and then that alerts the fetal medicine team that there might be a chromosomal or a single gene trait. So the first thing to say is that there is testing that can take place in pregnancies now, and they're much faster turnarounds of results, and often we can get a result back in 2 or 3 weeks now, either by looking at the chromosome number, and those results come back very quickly, in a few days, but also looking at what's called a clinical exome. So this means looking at the coding sequence of all 20,000 genes and comparing them between the unborn pregnancy and the parents to look for these traits which might be causing these congenital abnormalities. And this has certainly had a big impact on fetal medicine and the detection of these congenital abnormalities. So that is certainly improved. Remember that the coding sequences of our gene only equates perhaps to 1 or 2% of our genome. So we're still learning a lot about what some of those other aspects of the DNA do. And well, I think we might come on to that later. But when a child is born, a lot of people with these abnormalities spend years trying to see if we can actually find the cause of their abnormality. And whole genome sequencing has now come in to try and improve that diagnostic odyssey. And in fact, newborn screening programs with genomics are being introduced as well. So that obviously has the potential to reduce that diagnostic odyssey from several years down to several weeks. And I think getting a diagnosis can be extremely important, particularly for people with congenital abnormalities where they are worried about whether there's a chance that a future pregnancy might be at risk. What we often find is by the time a child with one of these abnormalities does get a diagnosis, a second or even a third child has been born potentially at risk of the same disorder. So being able to make that diagnosis early is important, both in terms of understanding the cause, but potentially in being able to look at therapies that may be useful for that child. So certainly this is one of the big improvements in genomics and our understanding of these rare conditions. These conditions in themselves may affect people only 1 in several thousand, but combined 1 in 17 people have one of these rare disorders, affecting at least 3 million people in the UK. So although they are unusual as a combined group, it is a very important set of conditions and a very common cause of neonatal death, unfortunately, between the chromosomal and these genetic disorders. So our ability to make these rapid diagnoses for children with either structural abnormalities, neurological abnormalities, or metabolic abnormalities, our ability to make that diagnosis is hugely improved. And that was underpinned by projects like DDD, Deciphering Developmental Disorders, which was an NIHR project, but also projects like the 100,000 Genome Project. Now, and I would urge people to, if they are interested in a particular topic, perhaps go to put into Google ODP for the NIHR, which will give them a list of subjects, and then you can search on things like genetics, and you'll be able to see for yourself what sort of research is available, and you can then do a find search on things like schizophrenia or congenital abnormalities. And for instance, we were able to find that there's 40 or 50 different types of projects which are looking at these types of conditions like schizophrenia, some of which are closed to recruitment, but you can very quickly get a feel for the type of research that is already out there and being carried on, and where the gaps are. And then if you see a gap, you can always go to your local clinical research network, or come back to us and say, well, what about this topic? And we can look into it for you.
Thanks, Julian. Thanks for that detailed answer. You quite clearly illustrated that we can detect conditions much more now, both in utero and after birth. What about treating them? So, for example, how advanced are techniques such as stem cell or gene insertion or other techniques? So, if you are picked up with an abnormality, whether it's a chromosomal or a single gene abnormality, are there any treatments now, or is it just a case of detecting more effectively?
So there has been a lot of progress recently with a new type of gene therapy type techniques, particularly for metabolic conditions and blood-borne conditions, because it's possible to take out some of the blood cells from the patient and then to manipulate those and then place them back in the individual. And the advantage of doing that rather than using a donor is that you don't have to worry so much about the immune system, because their big issue has always been, it's the same with cancer, is the big issue is we're always in fight between our immune system and then things that we feel are foreign within our body. So if we have a stem cell transplant, there's a concern that we may reject that from our bodies, and so there is a big issue in being able to dampen down the immune system to be able to introduce this. But there has been major progress. There is an issue, look at spinal muscular atrophy, a new drug has come in. Sickle cell, new drugs have come in, and for leukemias. But some of these treatments are incredibly expensive. So the most expensive drugs in the world are for gene therapies for young people. So the most expensive one being for hemophilia, $3.5 million for a single infusion. So over £2.5 million for one injection of a drug. So it's a huge amount, but real progress. Spinal muscular atrophy, a hope of many years improvement, and for sickle cell as well, the potential, at least it looks like, to be able to reduce the sickling crisis and reduce the amount of pain that individuals are going through. So it's coming, with regards to gene therapy, and the progress has been through something called CRISPR, and that is much more targeting of the genome to be able to place in genetic material that we're interested in more precisely into the genome, and rather than being more random. And the concern about random insertion is that, is it going to induce cancer by placing these genetic sequences right next to important on switches for cancer, and cell activation. And there has been concerns about that in the past. So that is improving, but these new therapies are coming in. But I think it's going to take a while before they become commonplace across the board. But certainly we've made a big steps in the last 5 years, I would say.
Brilliant. Thank you very much, Julian. Just as a bit of housekeeping for those on the call, if you could mute, press the mute button so any background noise is eliminated. Thanks very much. We'll go on to the next question. And this goes back to the issue around lifestyle and environment interplaying with genes. The question is, how do you minimize diseases such as diabetes and prostate cancer when they clearly run in families?
Right, it's a challenging array of questions tonight. So the first thing to say is, outside of these Mendelian disorders, so these single gene traits where the risks of diabetes, of cancer, could be really high. So in some conditions, for instance, BRCA1, the Angelina Jolie effect, which you maybe heard of, where the risk of breast cancer can be as high as 50% by age 50, 70% by 70, 80% by 80. And in BRCA2, for instance, a similar gene, the risk of prostate cancer can be extremely high, clinically significant prostate cancer risk of 25%. actually the majority of diabetes and prostate cancer will be one of these polygenic multifactorial situations where there'll be some genetic factors, but also environmental factors, sometimes of which we don't truly understand. So the vast majority of type 2 diabetes is felt to be partially related to modern living. So related to the fact that the mismatch between biological evolution and social evolution. So in biology, we don't know where our next food is coming from, and we don't know if that snapped twig outside means we're about to be eaten. So what we have to do is be on constant alert. We need to hoard sugar, salt, and fat whenever we see it, because we don't know when the next meal is coming. And we need to be live for hunting and gathering. Whereas in nature, supermarkets don't exist, and really that access to food in a regular fashion has only really come in the last few hundred years. Whereas modern man's been around for 200,000 years, and as I said, our genes 2 billion years. So we naturally, unfortunately, place ourselves subconsciously at risk of some disorders. We know, for instance, cancer is very much a disease of aging, and modern man life expectancy being in the 70s and 80s has exposed us to a number of conditions where there aren't evolutionary forces, because evolution tends to be impacting on whether we reproduce and are around to look after children. So in nature, takes into consideration people into their 40s and 50s, not so much 70s and 80s. And prostate cancer is a tricky one because of the rule of 85%, which is 85% of men at post-mortem. So after they've died, if you look at their prostate, will have abnormal prostate cancer cells. That's 85% of men over 85. So if you go looking for prostate cancer, you will often find it, but that doesn't necessarily mean that it kills people. And many men die with prostate cancer rather than because of it. So there is always a concern about over medicalization when it comes to these things. So what we're trying to do is to spot disease at an early, treatable, identifiable stage, but not to overtreat it where it's not going to cause a problem. So to do that, you've got to understand the condition extremely well. So again, this is where research is so important, to understand who is likely to develop that early and aggressive prostate tumor, which will catch them in their 50s and 60s. Not so much of a concern about picking up slow growing disease in men in their late 70s and 80s. And all we do there perhaps is run the risk of causing a lot of side effects like incontinence and impotence. So trying to work out effectively what is causing those sort of disease, but also what is influencing prognosis, how people will do in these types of conditions. So you know how to intervene, but also when to intervene. The answer to all this, I believe, is the integration of the DNA code with the postcode. We need to link different data sets together, and crucially, we need to match them to healthcare outcomes. We need to be able to understand the natural histories of these conditions and effectively develop equations for life. So how do we actually reduce the burden? Well, I think the first thing is we need to understand what's causing it. And there's a lot to be done still to understand, for instance, what's causing prostate cancer in men of African descent. So that needs further research. We know that genetic factors have a massive influence on all of these things. We know diet is important. We know, for instance, in prostate cancer, processed tomatoes and green teas can reduce the risk in the longer term. So can we in some way identify that more effectively? Can we offer screening to men with prostate cancer in a more acceptable way than we have now? So at the moment, biopsies cause infections. They're not very pleasant. And we know that PSA, for instance, blood test, which is acceptable to a lot more men, but it's not a perfect test. You can have prostate cancer with a normal PSA, and you can have a high PSA and not have prostate cancer. So we need better tests. So that's going to include urine tests. It's going to include liquid biopsies for cancer. And in the same way, we're going to need better ways to pick up changes in blood sugars. And obviously now with mobile phone technology, there are massive opportunities to pick up some of these diseases by self as well. But also better ways to track blood sugars moving forward, because what's crucial, we know through research in diabetes, is that if you can reduce the length of time that people are running high sugars, you're much better positioned to reduce the risk of problems to the eyes, to the heart, to the risk of stroke, and to the kidneys, and heart attacks in the longer run. So I think self-monitoring is going to be key. Mobile phone technology will be key. I think working with patients to understand what's going to be acceptable is key, and its integrated data is going to be important moving forward, to truly understand when best to intervene, because there is a danger of over-treatment and over-diagnosis.
Thank you, Julian. Can I follow that up with a bit more of a discussion around polygenic risk scores and the potential of people sending off a sample of blood and being told their own risk for diabetes or cancer or whatever, blood pressure, whatever it might be. How advanced is that? How useful is it? And perhaps you can touch on some of the big biobank type studies and how they might contribute to the knowledge and the capacity to do that type of thing in the future.
Thank you. The short answer is, at an individual patient level, I've got doubts at the moment as to how useful these are in isolation. I think it's used, for instance, to a degree for breast care. It's used a lot in cardiovascular risk, so risk of heart attacks and strokes. And it's coming in pharmacogenetics. So that's thinking about what dose of drug to give to people to reduce the side effects and to enhance effect. I think the pharmacogenetics is the most interesting of those three. And there's certainly a lot that we could do to make sure that patients receive not just the average dose for somebody of their age, but we know that everybody's got subtle variants throughout their genome, which are going to impact on how they respond to drugs. So we all know that some patients respond better to painkillers than others. We know that some people get problems with warfarin, blood thinning agents, or statins for their cholesterol and get muscle aches, and others patients are fine. And it's not in the minds. There will be genetic and scientific principles why we behave differently. And I think we'll look back in 20 years and say it was crazy that we were giving everybody the same drug, and we were trying to be equitable, but actually it was madness. And we'll look back and we'll laugh in the way that we laughed at leeches. We need to give people the correct medication for them, and standardizing treatment is not good. So I think that will work well. Polygenic risk scores for things like breast cancer should not be used in isolation. They should be used alongside things like alcohol intake, because we know that even a unit a day can increase the relative risk by 7%, which equates in real life to about a 1% increased risk per drink per day. But also body mass index. One of the most important factors in breast cancer risk is weight gain from the age of 21, but also taking into consideration postcode, and also linking in your family history and the results of any genetic testing. So it's bringing different data sets together that will be key. The problem is what do you then do with that data? And I always go back to the 2020 A-level algorithm fiasco, where A-level results were given not purely on attainment of the child, but also on the attainment of the school of the previous 5 years, effectively postcode lottery. So some children would be unable to receive the top grade, even if they never got a question wrong in 2 years, because of the attainment of the school. And so there is a concern that if we get more accurate results for the country, it may not fit into our sense of social justice. So I think integrating data for healthcare rationing is something that makes medical sense across populations, but it may not make ethical sense or political sense. In other words, will women that live in Rutland not receive healthcare compared to women that live in Leicester? Because we know the risks are higher. And are we really comfortable with that? It may be truly equitable, but I'm not sure we're quite there yet. And I think as a country, we need to have a debate around the coulda, shoulda, woulda aspect of healthcare. So I think that's the second point. The third point is about the accuracy. How much more does these subtle changes throughout our genome, if we look at them, make above things like our exercise regime, the food we eat, our body mass index, our family history? And I'm still not totally convinced that these are accurate enough to make big decisions on in isolation. And I would say perhaps the best guide to a lot of healthcare is what we eat and our psychology and what we drink and if we smoke, and our body mass index. And if you take all that on board and your family history, a lot of the time the molecular is still not as strong as those factors. And I think there is a concern that if we factor them in, people then lose a sense of control. And then anyway, if I told you you were at 5% higher risk of a heart attack or Alzheimer's, what are you truly going to do with that data? And I think that still needs to be unpicked, because
good point. And I guess we don't really understand the impact on psychology, mental health, anxiety of this type of information in individuals, do we really? That's the other unknown, I guess.
See, I'd like to ask you a question as a GP. If a patient were to come in with perhaps a commercial or a private test, which and perhaps has looked at these genomic profiles, and it suggested that they're at a 10% higher risk of cardiovascular disease or Alzheimer's, how does that make you to feel, and how can you use that as a real clinician in the real world rather than an ivory tower?
There's a number of issues as GP. One is then sort of explaining the issue of risk, which isn't straightforward sometimes, what it actually means. And we have that because we have cardiovascular risk scores, and sometimes explaining that to people as to whether they should have a statin or not is not straightforward. So I think we do need some training and some tools in terms of explaining risk to people so they can understand what it actually means for them. And then I guess it really is around, we're still at the level of trying to encourage people to adopt lifestyles that not only will hopefully reduce the risk in the issue that's been raised, but generally improve their health. So I think still many of the messages are generic around smoking, exercise, good diet. But I can imagine what my real fear is that people will become highly anxious when they get this data and want to do something quickly now, whether it's a pharmaceutical intervention or a rapid weight loss or whatever. They'd want to do that without realizing what the risk actually means. So that's really the concern I have. But as you say, this is a science which is developing, and the science how we apply to the public is also developing. And certainly healthcare professionals do need further training in it outside the clinical geneticists like yourself. Now, Julian, we've got a few questions which are around a bit more specific around what role genetics may apply in treatment. So we've had a question from somebody. It's quite a detailed clinical scenario of somebody with a brain tumor who suffers some nerve damage and has got foot drop. So in that sort of scenario, that clinical scenario or any other scenario where you get nerve damage, and as you know, nerve damage often is very difficult to reverse. Is there any potential for genetics or genetic therapy in this area?
So obviously, I'm sorry to hear about that for this individual. So the first thing is the role of genetic testing within the tumor. So when the tumor is removed or biopsied, whole genome sequencing could be carried out now, potentially to try and understand where the weaknesses are in with the tumor. So what we call the tumor drivers. So what is actually making those cells divide fast and out of control, not dying off at the appropriate time. So by understanding those genetic drivers, there is the opportunity to come up with more specific treatment. So that might be an immunological treatment, for instance, or a DNA repair based treatment. So we might be able to get a more targeted treatment based on that individual's tumor. What I call the Death Star analogy from Star Wars. You can't blow up the Death Star unless you've got the plans. So the second point is the dose of the drug, and also picking the right drug for the patient on how they are likely to respond to it. So we know, for instance, for bowel cancer, individuals now are not allowed to receive a particular type of chemotherapy unless they've had a genetic test first, because of a risk of major side effects. So I think we will see a lot more of this, where people will need to go through genetic testing, not just of their tumor or for their condition, but how they're likely to respond to the drug that they're given, because of the risks of side effects moving forward. And I think that's a massive step forward. We often think that genetics is all about risk. Well, actually, there's 3 R's. There's reason. Why has a family got the disease in the first place? There's response, which is how can we predict what will happen when we treat them? And we know a lot about that from microbiology from COVID, for instance, but it's certainly important in pharmacogenetics and oncology. But there's also the concept of risk moving forward, which is much more complicated, as you've discussed. And that really involves combining those 2 other factors, but also looking at large data sets of population. So I think we're going to need to look at more patients and the side effects that they've had, and try and understand what is driving those sorts of problems that people have had. It's possible in time stem cells might be able to be used to support people with neurological damage. It's a difficult field. They're also going to need, sometimes drugs can be used to reduce neuropathic pain. It's always important to be aware of allied health specialists such as physiotherapists, occupational therapists, and supporting aids that people may be given to support them with their conditions as well. And I think there's a lot to be said for thinking about the way patients can access these services. We've looked at something called a medical supermarket, whereby you invite people to an event. There may be 1 or 2 talks, but actually you have psychology, psychotherapy, smoking cessation teams, allied health professionals all there, and people can access them in the same ways that they do a supermarket. And I think we need to re-evaluate how we deliver some of these types of services, and I think we need to be more grown up in the way that we go in to receive so many of our services in a supermarket type model, and I think we need to think of new ways of working in the NHS, because it's so stretched, but there are novel and cheap ways of trying to help to support patients with these holistic type of problems. It's not just about events. I think we need digital solutions as well, and we're looking at digital solutions as well to help with self-navigation. A lot of people do use the internet these days, and I think there's a lot that can be done moving forward to support patients.
Thank you, Julian. Just on the point of cancer, we talked about breast cancer before, talked about neurological tumor just now. Do you just want to give us a quick summary in terms of which of the tumors we should be doing genetic testing for, which patients, in terms of where it can have greatest impact? I know breast cancer is one, but do you just want to elaborate what we ought to be doing now and what the research may help us to do in the near future?
Thank you. So the first thing is to say in bowel and womb tumors, we should be looking for genetic instability, which could be suggestive of an inherited condition called Lynch syndrome, which is really quite common. We think one in 100 or 1 in 200 people have probably got this condition. And the way I like to explain genetic instability is you may remember the 1980s, and we used to listen to a radio on a Sunday evening, and people would put a tape in the radio and try and record the most popular tunes of the week and wait for the DJ to stop talking, and then would press play and record and then predict when the DJ would start again. And in 1984, Shaka Khan came up with a repeat sequence in their song which went something like Shaka Khan, Shaka Khan, Shaka 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 Khan, Shaka Khan, Shaka Khan. So there'd be like 12 repeats of Shaka Khan in the song, but people would record perhaps 4 repeats or 8 repeats. And that ability to replicate the repeat sequences of DNA sometimes goes wrong in Lynch syndrome. And those repeat sequences are throughout our DNA. So we can measure the inconsistency in the replication of DNA sequences in the same way as you can in a song. And we can look for that genetic instability in a number of different tumors. So Lynch for bowel and endometrial is one. Different types of genetic instability is really important in ovarian cancer. So in the past, we would look at people's family history for ovarian and perhaps go on and then do BRCA testing and pick up maybe 10 or 12% of patients. But we now know that a number of patients have mutations within their tumor, which they've not inherited. But in fact, if you look at genetic instability, we now know half the patients have got genetic instability. And that's important because it opens up new drugs and people have much longer without their disease and do much better if they are on these drugs and if they're likely to respond. So we're moving almost to a post-genetic era where rather than purely looking at the genes changes which are inherited or within the tumor, we're looking at the impact on the whole genome, that's the whole 6 billion letters, by looking at particular signatures and particular changes which are occurring throughout the genome. And the problem is once you damage the genome in lots of different areas, those areas include other genes. So it's what we call the Swiss cheese model of risk. So the idea that the airline industry came up with, which is that there's no such thing as an accident. It's due to a whole series of minor changes which all line up on a particular day. And it's the same with cancer. We think cancer probably occurs when 6 or 7 of these pathways are interrupted in some ways. And it may be that, for instance, in Lynch, that the inability to replicate DNA sequences is one area which goes wrong, which then increases the likelihood of a second area going wrong, which then increases the likelihood of the third, fourth, and fifth until you develop a tumor. So I like to think of it as, yes, we're looking at genes which are inherited, but we're also looking at gene changes within tumors. But now we're looking at these genomic signatures, which are looking much more broadly at what's actually going on within the tumor. So you almost don't need to look at the individual genes themselves. You're looking at the impact of the genes on the genome. So whether or not that's going to put me out of a job or not, I don't know. But what's important
I'm sure it won't, Julian. If not, then you can take up a singing career, because obviously on this
well,
level,
how about that? Hey?
you can
How
sing
about
it.
that? I don't think I'll be making a lot of money if I do that. Yeah.
Okay, thanks very much for that. Specific question now around sickle cell. We've had somebody send in a question. When will gene therapy be available for sickle cell so we can sort of overcome this horrible disease?
Yeah, so it's a real quandary, sickle cell, because we've known about the genetic fault in sickle cell for half a century. And there's been lots of false dawns with sickle cell with regards to gene therapy and improvements. There has, in the last 5 years, because of CRISPR, been further developments and improvement in replacing the abnormal beta globulin gene and its production of the sickling effect on the hemoglobin, and which damages the structure of the red cells. So the red cells are the ones which actually carry the oxygen linked to iron. And what's so special about iron, which is held in the hemoglobin, is that iron is able to bind oxygen and then release it where the oxygen level's low. So it binds it when oxygen is high and it releases when it's low. And it just blows my mind how nature, over these billions of years, has been able to identify iron and capture iron and use it in this just truly mind-blowing way. So what needs to happen is you need to remove this particular type of protein produced by the beta globulin gene, and either by silencing or changing it. So with CRISPR, it's possible now to do that really quite effectively. The problem is either the immune rejection. The second problem, the concern is that are you inducing cancer by putting genes randomly into the genome and potentially stimulating the go faster signals in the cell called the proto-oncogenes. And the third problem is if you're doing this on the cells of the individual, so you don't need a donor, because obviously most people don't have a close match relative that they can use for stem cells. The concern is that you have to wipe out the bone marrow beforehand, which means you have to do something called conditioning, where you give chemotherapy. And the danger is that the conditioning can be so severe that it either causes the patient to have real problems with things like chemotherapy fog, where people feel just so washed out and it affects their brain cognition. There was an article about this only last week in the BMJ medical journal about the effect on brain functioning of chemotherapy. But also, does it biologically age an individual to have chemotherapy and generally make them weaker? But also, can it cause cancer in the longer term as well? So there is research now, not only about gene therapy for sickle cell, but also about using weaker forms of conditioning. And all of the concerns about that. So it is this conditioning type of research is ongoing. I think it's going to be a while yet, to be honest, till it will become mild enough but still effective. So I think it's going to involve multiple different therapies and then therefore involve multiple different types of research trials. And I suspect we'll be back to AI and big data sets as well. But it's crucial that the sickle cell community take part in this type of research to look at the conditioning effect. What's exciting about the gene therapy research is that it's the closest we may get to a cure for sickle cell. So if we can get conditioning right, there's a really good chance that these people may end up actually with long-term improvement in functioning and reducing the complications and the pain caused by the sickling effect. So really exciting research. Gene therapy is actually looking quite good. I think the biggest problem now is the conditioning of the bone marrow.
Sure. Well, that's very exciting, and hopefully will come to fruition in the not too distant future. We've got time for a couple more questions. So this one, if we could get a quick answer to this one, because it's something that I was quite intrigued by when it came in. This is the concept of junk DNA and what conditions may be attributed to junk DNA. Now, I don't know what junk DNA is, so perhaps you can explain to us, Julian, and are there any conditions associated with junk DNA?
So junk DNA really means the DNA that we don't necessarily know yet what it does. So we know that our genes are the ones which produce the proteins, the machines that do the work in the cells and provide the scaffolding in the cells. And we know that in between those coding bits, there are called introns, which are breaks. And they are important for telling the cell to get ready. And the way I like to think of that is a bit like an advert in a TV show. You've got your main TV show, and you've got adverts in between. So we've got that. But we now know the junk DNA, and that probably equates to about 98% of our genome. We've got 6 billion letters. That junk DNA is probably quite important, a lot of it, for gene switches. Knowing when genes are switched on and switched off, not just at a particular moment, but also different stages during our life. And we know that a lot of these things are involved in what's called RNA processing, which is the moving of the signals important in protein production throughout the cell. So the microRNA, the transfer RNA, and the ribosomal RNA. So we're beginning to understand that, but this is moving into the murky world of epigenetics, which is about when genes are switched on and switched off. Some of those traits can be inherited, by the way, and there's a lot of interest, for instance, in the impact of stress on pregnancy. So they're following up a number of women that were pregnant during 911 to look at the impact on their children and on their grandchildren. Because remember that the eggs for your grandchild are carried in women, because their daughters will be producing their eggs when they're in utero. So you've got 3 generations in one. It's not 2 generations in one in a pregnancy. You're actually carrying 3 generations in one. And there is a concern that those genes will be gene switches are impacted by stress and environment and by things like famine. So there's a lot going on there that's still not understood. But I suspect some of it will be remnant DNA and won't have a role. But I suspect we're only at the beginning of truly understanding what a lot of this junk DNA. And it won't be junk. So I'd be quite nervous about having 98% of my genome taken out.
Yes.
But remember, we share 50% of our genome with a mushroom. So you know, we have a lot in common with nature, and we're all connected.
Yes, I guess it's the genetic equivalent of dark matter in physics, isn't it? I would guess.
Absolutely, yeah.
Anyway, we're nearly at the end, but I'm just going to finish off with a bit of an intriguing question, which I hope you can have a good stab at answering. Genetics clearly has progressed rapidly in the last decade, in the last few years in particular. Where do you think we'll be, first of all, in 5 years? Because most people can imagine that fairly easily. And maybe in terms of crystal ball thinking in 20 years, what do you think the role of genetic therapies will be in medicine?
So in 5 years, if you're over the age of 50, I would like to see everyone having a pharmacogenetic profile. So if you're picking up your statin or your blood pressure pill or your painkiller, you'll be able to make sure you QR code that in your pharmacy to get the right drug at the right dose. I'd like people with rare diseases to have a dedicated electronic patient record and a named physician and support to take them through. So that for 1 in 17 individual, and that whole genome sequencing would become something that would be available to a lot more people much earlier on in the process, and that we would have started to tackle some of those disparity issues in terms of having genome analysis from a much broader background of people as background to then to be able to compare with patients. So that's in 5 years. 20 years, we should be at that integrated data science point where we should be able to give people much clearer sense earlier on if they want to know where they're going to be at with regards to what particular tumor risks or cardiovascular risks, so we can screen people based on risk rather than age alone. As to whether or not people want those results, are happy to give the data for those results, and whether they will act on those results, I think is still all up for grabs. But 5 years, much better with regard to response and patient true involvement, engagement, and practicality. 20 years, I think we'll be dealing much more with risk. Oh, sorry, I can't hear you. I don't know if it's me.
Yes, I was muted. So yeah, thank you. That brings us to 8 o'clock and the end of our webinar. Thank you so much, Julia. You've answered a range of questions in a very expert way, and really thanks to you for your time and for answering the questions, and also for the people who sent those questions in. I hope you found it an insightful and informative session. Thank you also to our hosts at the NIHR Research who've set this webinar up. If you want to learn more about research and how you can take part in studies, even if you're healthy, search for the Be Part of Research website. That'll give you loads of information of potential studies you could take part in. As I said previously, this webinar will be recorded. So we'll send a recording to everybody, and also an evaluation form. So please take a moment to complete that. We want to run more of these events. So we want to know what is right, what went well in this webinar, and what we could improve. So it just remains for me to thank you all again, and hope you have a very pleasant evening. Thank you.
Thank you very much, Azar, for the opportunity.
Thank you, Julian.
Thank you.